Welcome back. We're going to move on to the urinary system now. So, gross anatomy wise, this system is fairly straightforward. You have two kidneys, two ureters, you can see right here or right here, that feed into a urinary bladder and one urethra. So, the majority of the anatomy that we're going to cover here is going to be internal. So gross anatomy, pretty simple. Two kidneys, two ureters, one urinary bladder, one urethra. Function-wise, the kidneys are very important in homeostasis. So these first three functions all say regulate, which means that they have a job maintaining the proper range, the homeostatic range of these three things. So the total amount of water, the total volume of water in your body, and the osmolarity of the different fluid compartments is going to be controlled by the kidneys. It's going to regulate the ion concentrations in the ECF. So how much sodium, how much potassium, how much calcium, all of that. And it's going to play a role in regulating pH. So the acid-base balance overall in your body is going to be maintained by the kidneys primarily. So we talked about the respiratory system's role in maintaining pH, and now we're going to get into the urinary role. In addition to the homeostasis, we also have the excretory function to get rid of waste products, foreign substances. We are producing the hormone erythropoietin and the enzyme renin. So we've talked about both of those. We use our kidneys to convert vitamin D into its active form, which is called calcitriol. And then during times of fasting or starvation, your kidneys can actually undergo a process called gluconeogenesis. If we break that word down, gluco means glucose, neo means new, and genesis means start or beginning. So what this means is that the kidneys can actually make new glucose. They can take other types of molecules, so fatty acids, they can take proteins, amino acids, and they can convert them into glucose. So the only time that we would do this is if your glucose stores were completely gone, because essentially we begin to catabolize our own body at that point. Okay, so location-wise, here is our digestive system, which we haven't gotten to yet. Here's the posterior body wall. Here's your vertebrae here. And the kidneys are sitting back here. So they're closer to the posterior wall than they are to the anterior wall. So that term, retroperitoneal, means that it sits behind this peritoneal membrane that lines the digestive cavity, so the abdominal cavity. So retro means behind, so it's sitting behind here. They are sort of kidney bean shaped. They have an adrenal gland on top of each one of them, not sharing any real function, just an anatomical location. Unlike all of the stuff we've talked about to this point, we do not have a serous membrane surrounding our kidneys. We have a sort of 3 layers of protection for them, starting with the outermost layer, which is indicated in green here. This is the renal fascia. So this is connective tissue that is going to sort of blend into this peritoneum and to the surrounding tissues, and it's going to hold the kidneys in roughly the right place. Now, when you go from laying down to standing or something like that, there is a few centimeters of give within the fascia, so the kidneys can move a little bit. Then inside of that, we have a perirenal fat capsule. So this is not just given no justice on this diagram. It is much thicker than this. It's a big, heavy-duty layer of fat that is surrounding them. It is designed to be protective. With the close proximity of the body wall here, the kidneys are fairly vulnerable to trauma, so we need to be careful and try to sort of give them whatever protection we can. So we've got this nice thick layer of fat there. And then the closest to the surface is the kind of this dark layer, brown layer right here, is the renal capsule. Unlike a serous membrane, this is very, very tightly bound to the surface of the kidney and is not designed to provide any friction-free movement or anything like that. It's designed to almost like saran wrap the kidney with this heavy-duty layer of protective tissue. So all of these are there to kind of surround and protect the kidneys as best as possible. Internally, we've got a whole bunch of anatomy. So this is where the anatomy really comes into play. The nice thing about the kidneys is the real kidney looks very similar to the drawing. So not all organs actually look like the drawing, but this one does. Okay, so we're going to divide this internal structure into two categories. The first is going to be the functional tissue, where we actually go through the process of filtering blood, we make urine, we deal with all those homeostatic mechanisms. That's going to be called the parenchyma. Parenchyma is a generic term for functional tissue. So the renal parenchyma are going to be the tissues involved in actually doing the job of the kidney. The other tissues are going to be collecting structures. So nothing is, they're basically just conduits to get the urine from the parenchyma through the kidney and then out of the kidney. So we're going to start with the functional tissues, the parenchymal tissues. There's two different types of tissue found in this category. The first is what we call the cortex tissue. So it's running all around here and in these pieces right here, which are called the renal columns. So this tissue is all cortex tissue. And then the other type is the medulla tissue, which is formed into these pyramid shapes. So these are called renal pyramids or medullary pyramids, but that's your two different types of tissue found in the parenchyma. So we've got the cortex, the columns, and the medulla, and the medulla is arranged into these pyramids. That is where all of the function takes place. From there, we move into the collecting structures, starting with minor calyces. Each pyramid has a minor calyx surrounding it. Minor calyx means small cup. And so as urine drips down out of these pyramids, it's going to collect in these minor calyces, which feed then into major calyces, which feed into this expanded area here in the hilum called the renal pelvis. The renal pelvis then hooks up with the ureter, and that's going to take us on to the urinary bladder and then out the body via the urethra. So once the urine hits these collecting structures, no more changes can be made. So whatever the concentration, whatever the contents of the urine, the water volume, once it's out of the parenchyma, we can't do anything about it. All this is conduits to move it out of the kidney. If the job of the kidney is going to be to filter the blood and maintain homeostasis, we're going to have a fairly decent blood supply. And we do. So here what we see is, first of all, our renal artery branches directly off of the aorta, which means it's still going to have a high pressure. That then branches into what are called segmental arteries. If you sort of mentally divide the kidney into 3 to 4 segments, each would get a branch of a segmental artery. Then we get to interlobar. So first let's define what a lobe is. One lobe of the kidney is defined as one pyramid and the area of cortex that overlies it. So that is a lobe right there. This would be a lobe right here. That is a lobe right there, etc. So interlobar, inter means between. So interlobar means between the lobes. So these are your interlobar arteries that feed into these guys right here that are arcuate arteries. They arch over the top of the pyramids. That then branches into cortical radiate arteries, which you can see here radiate out into the cortex. From there, we're going to go into microscopic vessels that we will talk about in a minute, but we're going to branch into what's called the afferent arteriole. Afferent means going into. And then we go into the glomerulus, which is a capillary bed, into the efferent arteriole. So right there, you should see something different. Generally, we go from an arteriole to a capillary to a venule. And here we're going from an arteriole to a capillary to an arteriole, which means that this capillary bed is still going to have a huge amount of pressure because we're still in the arterial system. Then we branch into a second group of capillaries, either the paratubular capillaries or the vasa recta, depending on what type of nephron we're dealing with. And from there, we move into the venous system. So this is a portal system where we've got a capillary feeding into another set of capillaries. Now we do have an arteriole between them, but remember we talked about this in the circulatory system, and this was one of the ways that we could arrange this. So capillary to capillary and then to vein. When you get to the venous side, we basically trace this pathway back out. So these are your cortical radiate veins, which lead into the arcuate veins, which lead into the interlobar veins. The only difference is that we don't have segmental veins. I don't know why. The interlobar go directly into then the renal vein, which feeds directly into the inferior vena cava, which obviously is going to feed into the right side of our heart. So this flow is important. You're going to need to know this so that you can figure out where you are when we begin to look at these microscopic structures here. So I just said something about nephrons. Nephrons are the structural and functional unit of the kidney. So this is where we do all of the work. There's over 1 million per kidney. So if you look at the cartoon on the first page, it's talking about kidney donation. And kidneys are something that you can donate while you're still alive. You can donate one of your kidneys because you have more than enough nephrons in one kidney to deal with your body. So it's something that we almost have a spare of. So this is where we're going to filter the blood and make the urine, is in the nephrons. If we look at them, we can see, first of all, going into some of this blood supply that we talked about before, one thing you want to get used to is understanding that the arcuate arteries and veins are the dividing line between the cortex and the medulla. So the cortex is above, the medulla is below. So this is how you orientate yourself to what you're looking at. You look at the arcuate arteries and veins and you know cortex is up here, medulla is down here. Okay. So when we were talking about the arcuate artery, we said we would come in on the arcuate artery and then we would branch into cortical radiate arteries here. And then we would go into an afferent arteriole, which means going into something. And what we're going into is this glomerulus, this ball of capillaries here. And then an efferent arteriole that comes out. So afferent means going in, efferent means coming out. And then it's going to branch into one of two capillary networks depending on what type of nephron. If it is a cortical type nephron, it branches into this paratubular network. If it is a juxtamedullary nephron, it branches into this network called the vasa recta. And then we feed into our venous supply. So here's our cortical radiate vein. We've got cortical radiate veins over here. The vasa recta often skips the cortical radiate and goes directly into the arcuate. And then, of course, we go into interlobar and then renal vein. So you really need to work on the blood supply so that you can figure out where you are when you're looking at this. Now on to the nephron part. We can see that the nephron starts with these capsules, okay, right here. and then it's going to feed into this tubular network that's then going to feed into a loop that comes back into a tube, and then multiple nephrons will feed into one of these structures called a collecting duct. So the capsule up here is what is known as a corpuscle. Okay, so the corpuscle is the capsule plus the capillary bed inside of it, and then the rest of this, which is termed the proximal convoluted tubule, the nephron loop, and then the distal convoluted tubule is what's just known as the renal tubule. So let's look at this piece up close. Let's start with the corpuscle. So this is what we're looking at. So this is the capsule, and then this is the capillary bed coming in. This is the afferent, and this is the efferent. You can see there is a size difference between them. And then the capsule leads into the proximal convoluted tubule. So the glomerulus is a ball of fenestrated capillaries. So when we talked about capillaries, we talked about 3 different types. And we said fenestrated, we're going to have these pores in the cells that we're going to facilitate filtration and make it happen much more efficiently. So these fenestrations are going to allow pretty much everything to pass through except for the blood cells and the major plasma proteins, the big plasma proteins like albumin. Now, the capsule that surrounds it is continuous with the tubule, and it has a parietal and a visceral layer. The parietal layer can be seen right here. It's simple squamous epithelium. Okay, it comes around like this. And then the visceral layer is made up of these cells called podocytes that actually wrap around the capillary loops. And they're going to help form what's called the filtration membrane that we're going to talk about here in a minute. So our capsule or corpuscle leads into the tubule. It's really, if you stretch it out, only about 3 centimeters, but has 3 different regions. The first region is the proximal convoluted tubule. It's going to consist of simple cuboidal epithelium with a whole lot of mitochondria and a whole lot of microvilli. So if we go back to functionality, mitochondria says, well, if I've got a lot of them, I must need energy. And we do. We have a huge amount of active transport that's going to happen here. Microvilli always means an increase in surface area. So I am increasing surface area for a process called reabsorption, and I have a lot of mitochondria to help drive that process. Then we go into the loop, which we have descending limb that goes down into the medulla, and then an ascending limb that comes up. We're going to see that there are thick and thin segments. Thick segments are simple cuboidal, thin segments are going to be simple squamous. Essentially, the thin segments are going to allow water to pass a lot easier, whereas the thick is not going to be permeable to water. That then leads us into a distal convoluted tubule where we have simple cuboidal epithelium, but we don't see the crazy high microvilli or mitochondria. So that indicates that we're not going to be as busy in the DCT as we are in the PCT. Distal convoluted tubules then are going to feed into collecting ducts. Again, a collecting duct does not belong to an individual nephron. It's not part of a nephron. It receives the distal convoluted tubules from many nephrons. So here we can kind of walk through this process. So this is what we're looking at. So you can see in the cortex is where we're going to find our corpuscles. Okay, so here's our dividing line here. Okay, so all corpuscles are found in the renal cortex. All of your PCT and DCT are found in the renal cortex. The nephron loop starts in the cortex, comes down into the medulla, and then comes back up into the cortex. And collecting ducts run the entire length, cortex and medulla. So here's our blood coming into our capillaries, and we are going to filter. We got those fenestrated capillaries, and whatever gets filtered out of the blood is going to be called the filtrate, and it's going to move through the proximal convoluted tubule and then down into the nephron loop. So here's a thick segment, and then we go into a thin segment, and then back to a thick segment. And then we feed into the distal convoluted tubule, which feeds into one of our collecting ducts. So the two types of nephrons, we have what's called a cortical type and a juxtamedullary type. So cortical are about 85%. They lie almost entirely in the cortex. They have very short little nephron loops that dip into the medulla. They are the workhorse of the kidney. They filter blood. They form urine. These will be associated with the peritubular capillaries. The other 15% are juxtamedullary. They are going to originate much lower in the cortex. The corpuscle will still be in the cortex, but it's going to sit much lower, like closer to the border. These have the very long nephron loops. They do the same thing as cortical. They filter blood, they make urine, but they have this third job, which is incredibly important. It's so important that it's the reason that we can live on land and not have to live in water. So it's a pretty significant evolutionary step that we took when we got these nephrons. Their job is to maintain a salinity or a salt gradient in the medulla so that urine can be concentrated when we are dehydrated. So we're going to talk about that a little bit later. These will be associated with the vasa recta. So we said that the efferent arteriole would either feed into peritubular capillaries or the vasa recta, depending on the type of nephron. So here you can kind of get a picture of this. This is a really great SEM shot that shows the actual balls of capillaries. So this is what the glomerulus actually looks like. You can imagine that there's going to be a lot of pressure in there because those vessels are just wound up so tightly. So here's a cortical type, okay, peritubular capillaries, very short nephron loops. And then here is a juxtamedullary type. Corpuscle is sitting much closer to our border, which again is made up of the arcuate arteries and veins. And then we've got this very, very long nephron loop surrounded in the vasa recta. The three sets of capillaries that we've talked about, the glomerulus, specialized for filtration. In order to drive only filtration, it is both fed and drained by arterioles. If you want to think arterial, it's in the arterial blood system, which means it has pressure. So we don't want reabsorption to occur right here. All our job is to filter. So in order to maintain filtering only, we have to maintain very high pressure. The peritubulars wrap around the tubules and are there to absorb the water and solutes that we need to get back after filtration. And the vasa recta consists of these very long, straight vessels that are going to play a role in that concentration of urine. Last thing to talk about anatomically is what's called the juxtaglomerular complex. This is an area where the distal ascending loop, okay, so basically the far end of the loop as it comes back up into the cortex is going to lie right up against the afferent and efferent arterioles of that nephron. This is where the kidneys are going to get involved in blood pressure. So we know that the kidneys are involved in this. We studied that role. We studied the renin-angiotensin-aldosterone system. So we know that that is going to come into play here. So this is where we get into that. So here you can kind of see what we're talking about. So here's the afferent and efferent arteriole, and then this is the proximal convoluted tubule, and we go down into the nephron loop, and so the nephron loop comes down, and when it comes back up, it's going to hit right here. So that's what we're looking at. This is the nephron loop. It comes and it is in contact, physical contact, with the afferent and the efferent arteriole. We're going to find three specialized sets of cells here, starting with these guys, the macula densa. So they are actually embedded in the wall of the tubule. Their job is to monitor how much sodium chloride is still in the filtrate after we've gone all the way through the PCT and all the way through the nephron loop, how much sodium chloride is still left. And what they're doing to look at this, what they want to find out is how fast is the filtrate moving. So the filtrate starts in this corpuscle here. And we know that filtration is driven by hydrostatic pressure of the blood. So the more pressure is in the blood, the faster the filtrate is going to move through the nephron. And the faster it moves through the nephron, the less sodium chloride is going to be reabsorbed into the body, which means by the time we get here, there's still going to be a high concentration of sodium chloride. So what we're doing is we are making an estimate of systemic blood pressure based on speed of the filtrate, and we're measuring that by how much sodium chloride is left. So when we have a high concentration of sodium chloride here, it's going to indicate fast moving filtrate, which indicates high blood pressure. The opposite would also be true. If the filtrate gets here and is very low in the concentration of sodium chloride, it means that it has moved so slowly through the nephron that we have pulled a ton of sodium chloride back out, and it's only going to be able to move slowly if blood pressure here in the glomerulus is low. So we are estimating systemic blood pressure by monitoring sodium chloride. So that's what our macula densa cells do. The granular cells, these bluish cells right here, are mechanoreceptors that directly monitor blood pressure and will secrete renin to start the RAS system as needed. Finally, these sort of long purple cells that we see through here, these are called mesangial cells. Their job is to kind of fill in the spaces between all of these pieces, and they contain gap junctions that are going to allow signals to pass very quickly from one piece to the other so that communication is very efficient here. We know that we need to make split-second and actually even fractions of split-second decisions to maintain systemic blood pressure. So it's important that we're able to pass signals very, very quickly through this entire complex.